Hey there. So this next idea is all about combining genes from different organisms, or inserting a new gene, or removing a gene or a gene version or allele from an organism. So what do you call an organism where you are placing a new gene or a new version of that gene into an organism? A gene that has never belonged to that species before. And we have technology now that allows that. As the screen suggests right here, we have cats that can glow, eggs that are anti-cancer or medicinal. We have a scorpion cabbage here. And we have eco-cleaning trees. All four of these examples, something has been done to them. They've been modified genetically to produce these unusual phenotypes or appearance. At this time, the slide wants you to hypothesize why scientists insert foreign genes into organisms. So, why do you think they do it? And here you go. The next task, number 2, says create a GMO, or a genetically modified organism, for the scenario below. So pick one of these topics: cuteness, making money, or the military. Create a genetically modified organism, or select an organism, any type of animal or plant, microorganism, whatever. Select one and change it in some way that you think will enhance or increase its cuteness. It'll make someone a profit, or it could be used by the military. So create a story or a scenario where that is possible. Alright, let's move on to the next slide here. This slide here appears to be a review slide. So check with your Chapter 13 notes 1 or 2 if you want to kind of understand some of the activities or tasks on this slide. But it's put here for a reason. Restriction enzymes play a strong role when making a genetically modified organism. It helps to break bonds between DNA molecules in specific regions. So you can get sections of DNA, aka a gene, and you could remove it from one organism in order to place it in another. These links right here, these 3 active URLs, you should go to, check it out, and it would help remind you about the possible benefits of cutting certain sections of DNA out, or maybe taking sections and combining them with another organism's DNA, etc. Alright. Gene splicing is, as the definition says, the joining of different DNA fragments. And it says restriction enzymes cut both DNA fragments and assist in their fusing. So here we have an original piece of DNA, or DNA from a foreign organism, and then part of it's getting cut out. That's the restriction enzymes, right? And the restriction enzyme can also help insert that new piece of DNA into another organism's DNA, or another individual's DNA. So it could be across species or within the same species. But think of each section as its own gene or gene version, aka allele. Alright, let's look at the tasks. It says the nucleotide molecules of DNA have charges that attract certain other nucleotides. So the reason why DNA strands can bond to other DNA strands, the reason why DNA itself even holds together, is because the atoms and the molecules that make this large structure, or this polymer, have charge attractions. And so what you need to do is kind of just study the photo and think of what might be the charges of the ends of this DNA piece right here that allows it to attract and bond to another piece of DNA from another organism or species up top here. So what is it happening between the edges where they bond together that allows that to happen, right? And it's just wanting you to think about, on the molecular level, what is needed to make a chemical bond, and that is these attractions. And here you have positive and negative charges, and just kind of study it and examine, are these arrows moving towards one another? Does it look like they pull each other or have some level of connection, right? In your response, you need to use the word charge attraction and the word bonding. All right, number 2 is identify and interpret what occurs to the image to the right. So the image to the right is this picture right here. And it's a model that a scientist has created showing a bacteria. And the bacteria has 2 forms of DNA. They have this loose coiled DNA here. And then they have this round DNA. And this is unique to bacteria. It's called a plasmid. And this plasmid here is interesting with bacteria because it's a form of DNA that they use, and humans can also do things to it. So your idea is to look through this, read the information that accompanies this model. All right, pay close attention to this black zone, and know that in this original version of the photo it didn't have it, but later it has it here. And then this arrow means it's being moved. Notice the shape of this. This is not the same shape cell as this original cell or bacteria. And, of course, right here. So this is the end right here. So the order, it goes from top down, follow the arrows, follow the language, and the information it's providing. All you're doing is to interpret something is to look at it and make sense of it and explain it in your own words. That's all you do. You look at the photo and explain it in your own words. Here you have URLs that you should in fact go and observe to make sense of the definition, this idea of gene splicing. Plasmid, as we just mentioned before, is a small ring of DNA from bacteria, and it's a very common source of DNA that can be altered, where you can insert a new gene or gene version, allele, into it. As this, so it says interpret a scenario and summarize using terms. So there's terms: restriction enzyme, bacterial plasmid, foreign DNA. That means DNA from a new source other than the organism itself, and gene and splicing, right? So it's saying use these terms to summarize what's occurring here. So basically what it wants you to do is use academic language, use the right terms to describe this process of taking a bacteria's plasmid and inserting it back into itself, but including a new section of DNA into it. So you get a foreign piece of DNA, or a new section, to combine with the older section, right? There you go. It says observe image below. Here's an eyeball. It says hypothesize purpose of using plasmid and gene splicing. So imagine this idea of inserting a new gene or a section of DNA into someone's eye. And why do you think they might do that? So hypothesize it. Someone might do that to blank, or for the purpose of blank. Alright, here a vector. What's interesting is that there are organisms that we can use, virus or bacteria. Bacteria we've talked about. We mentioned plasmid at their DNA. But we value them because you can modify bacteria or viruses to insert a gene of your choice or a section of DNA of your choice into another organism. Just like viruses and bacteria effectively infect a person and get in there and can cause damage to someone's cells. That's what having a cold or being sick is they're infiltrating your cells. They're getting to your cells, right? But what you can do is you can change and modify a bacteria or virus. You can give it new DNA sections so that it's harmless, but it can still get into your body and get to your cells. You want them to get to your cells because that's where they're able to insert a new gene or allele into that cell so that that new gene or allele can get to the DNA of that cell, and that cell will use that new DNA hopefully to help the body. The purpose why scientists or doctors are doing this is that it inserts new DNA to help someone. A perfect example of this is this website. And this website is called Engineer a Crop. And it comes with an activity worksheet that I'll provide to you. Just remind me when you get here. And the idea is you go to this website and you're going to study how a carrier organism, or a vector, and in this case I think it's a bacteria, is used to put a new gene into a plant, and why. So what about this plant, okay? So you can go to the website, check that out. Also know that there's this wonderful website here from Learn Genetics about gene therapy, and it shows how a virus is used to insert a helpful gene or a helpful gene version, an allele, into a human being to help them as well. So I recommend you doing this activity. Of course, it's part of the task. You need to go to this website and follow it and follow the worksheet as you go through it. But you also should check this out and see how it can help humans. Let's keep this moving. All right. So this is straight out of the textbook, and it's an example of restriction enzymes doing their work. Now, your job is to fill in this table right here based on which enzyme you'd need. So different restriction enzymes cut different sections of DNA. So if you had this section of DNA and you wanted to cut it, and you wanted to remove part of the DNA from another, you need the restriction enzyme. In fact, you need one of these. So you have to look at enzyme D, E, or F, and you have to decide which one would help separate this strand. Enzyme D cuts between A's. Is there two A nucleotides together here? If so, it might be the right choice. Enzyme E can cut between two G's that are parallel. You have to look along here. Are there two G's on either side of the where the nucleotides are there 2 G nucleotides next to one another? If so, use it. Enzyme F will cut or break bonds between G and A nucleotides. Are there a G and A nucleotide on the sides here? If so, you're good to go. But when you decide and pick an enzyme, you do it based on the side of the DNA. So you don't go back and forth here. It's not C to G here. It's C to A on the top, or G to T here. You only study, when looking for where an enzyme might be able to cut or break a bond in DNA, you have to study this side walls here, not back and forth between the two attracted to each other. It's the nucleotides on each side. That's what you study and look at. It says draw on the action of each enzyme. So use this top filled in one to know what to do here. So whichever enzyme you pick, it's going to cut the DNA on the side here and on the side here. And you list what enzyme you're going to use, what restriction enzyme, and then just like this dotted line here, there's a dotted line that shows it's cutting between the G and the G, and then it cuts it between the G and the G on the wall here. You need to show just where it's cutting in this box, and then show the two strands separated. So if it cuts it there, if it breaks that bond, it separates the DNA. So draw what it shows here, just like here. All right, good luck. All right. This is building off that same idea. It's referencing this table and where you broke down what a restriction enzyme is doing. And it says, use the example provided in the data table to illustrate a single-stranded dangling end of DNA. So when a section of DNA is cut with a restriction enzyme, it leaves an open end. This is called the open end right there. It says AATT. So it's these nucleotides that don't have a bond on the other side. It's a short section of single-stranded DNA. Same here. Short section of single-stranded DNA. It says, are restriction enzymes very specific as to where they cleave DNA? So the question just asked, are restriction enzymes specific to where they break bonds? Or can a restriction enzyme break any bond anywhere in DNA? And so it just says, explain your answer and give an example. So use this information from the table for you to write a response to that. Are restriction enzymes specialists? They'll only break bonds in certain areas? Or can restriction enzymes, can you use one to break a bond anywhere along DNA? All right, finally, there's this site from learngenetics.utah.edu, and this worksheet goes with it. However, I will make a separate video to help you here, and just so you can see the website and see how the worksheet goes along with the worksheet. Okay. All right. Good luck. That's it. Don't worry about slide 10. And we're good.